So we have some genuine talk now. You will put me on, allow me to host. So when you all are coming to the campus, Ayush?
Ma'am, I'm not coming to campus.
Why Ayush?
Ma'am, I've given admission in some other schools. That's
Okay.
why.
Okay. And then who are coming then next? Jaydeep. Jaydeep, can you hear me?
No, ma'am.
When is your SPI interview? SPI interviews plan in the recent dates?
Ma'am, it is coming soon. SPI.
Have you got the date of interview?
No, ma'am, not yet.
Not yet. Not yet. Okay. Vaibhav, when you are coming? Vaibhav Gusare. Vaibhav, when you are coming to the campus? Vaibhav, can you hear me? Hello. So today we'll start the next phase, that is metaphase 1. Got it? So can you see this figure, what happened in metaphase? This one is the figure of metaphase, that is homologous pair. Can you see the figure? Wait a minute. Wait a minute. Okay. So we have completed this stage, that is dikinesis. What happened during dikinesis? You can see here. Can you hear my voice? Students, Ayush? Vaibhav? Okay. So what we have completed in dikinesis, that is during this phase, chromosome continue to condense and short. First of all, that particular chromosome will become condensed and it will become short. The separation of homozygous chromosome proceed at the chiasmata, get shifted towards the end of chromatid. मैंने आपको जो बताया X shape structure जो आता है, X shape structure जो होता है वो एक दूसरे से separate होना शुरू हो जाता है. They start separating. So that particular process we call it as a terminalization. उसको क्या बोलते हैं हम? We call that particular process as a terminalization, that is called as terminalization. So you can see that is, you can see here that is called as terminalization. The nucleus and the next event which is important in dikinesis is that nucleus and nuclear membrane completely disappear and kinetic spindles become apparent. वो जो kinetic spindles होते हैं, spindles आना शुरू हो जाते हैं और जो nuclear membrane है that will completely disappear. That will completely disappear. Now we'll see the next stage, that is metaphase 1. That is metaphase 1. What happened in metaphase 1? First thing you can see, this is the metaphase 1. These are the centrosols. ये बीच में जो होता है उसको हम क्या बोलते हैं? At center, this is present. It is called as centromere. उसको बोलते हैं centromere. ये दोनों सेम है, मैंने आपको बताया. Homologous pair. Homologous means what? What do you mean by homologous? The particular chromosomes which are genetically and structurally similar, we will call it as a homologous chromosome. So these two are the homologous chromosome. One is blue and one is, suppose for example, one is blue and one is yellow. What is the center part, round part, this one we call it as a centromere. ये जो साइड पे आते हैं उसको हम बोलते हैं centromere. That is called as centromere. One will move toward north pole and south pole. These centromeres, yes, centromeres, what is the use of that centromeres? First of all, the rays-like structure which are formed, this one, we call it as a aster. Aster. The lining structure, you can see this one. These are called as spindle fibers. What it is called as? Spindle fibers. And the outside one, it is called as cell membrane. Cell membrane. So what is the speciality of metaphase? As that, like that, in any stage, whether it is in mitosis or meiosis, what happens in metaphase 1? The spindle formation is completed. First of all, this spindle, जो पहले होते हैं डाइकाइनेसिस में क्या होते हैं, ये थ्रेड लाइक होते हैं, ऐसे पूरे डेवलप नहीं होते, पर मेटाफेज में दे फुल्ली डेवलप, दे आर फॉर्म्ड फुल्ली कंप्लीटली, and the bivalent move. Why it is called as bivalent? You can see this structure, we call it as a tetrad also or bivalent also. This bivalent move and arrange themselves at the equatorial plane. ये center में जो होता है, this one is the center. So whatever, now here for example only 2 chromosomes are shown, but there are many chromosomes, all will get arranged at the center. You'll get second chromosome here, third pair here, fourth pair here, likewise. For example, we have shown just one pair. Got it? So this particular chromosome, they will get arranged at the equatorial plane. Equatorial plane is nothing but center. Center में आके सब chromosome लेट जाते हैं, उसको हम बोलते हैं metaphase 1. But what is difference between mitosis and meiosis? In mitosis, only single chromosome come, all the single chromosome come and get arranged here. But in meiosis, what is difference? Pair of chromosome. This is the pair of chromosome which are, in which chromosome recombination has taken place, exchange of genetic material has taken place, and after that what ये सब कब होता है? Exchange of genetic material, recombination, that we have already completed in prophase 1. So after the exchange of genetic material सब हो गया, अभी इसका chromatid, ये green वाले जो blue वाला है उसके genes yellow पे आ गए, yellow वाले के genes blue पे आ गए, ये सब complete हो गया और अभी वो center में आके लेट गए. That is the pair get arranged at the center. Next point is chromosome fibers. Chromosomal fibers, whatever chromosomal fibers are there, extend from pole to the centromere of the corresponding homologue. जो chromosomes fiber होते हैं, आपको मैंने पिछली लेक्चर में भी एक बताया कि यहाँ पे एक स्ट्रक्चर होता है. Here at the centromere, you can see here one structure is present. That particular structure, we call it as a, here one particular structure is present here. That is called as kinetochore. ये बात हमेशा ध्यान में रखना. This type of questions are always asked. That is, ये जो spindle fibers है, ये जो lines आप देख रहे हैं, वो कहाँ पे अटैच होती है? हमको दिखने को क्या दिख रहा है यहाँ पे? They are attached to the centromere. This one. They are attached to the centromere, means this round structure, got it? This one. But it is not like that. Outside of that round structure, suppose this is our chromosome. This is our centromere. To this centromere, one such type of structure is present. We call that structure as a kinetochore. उसको क्या बोलते हैं? Kinetochore. To this kinetochore, these spindle fibers are attached. From this also one kinetochore is present. That you can't look, and to that kinetochore, in this type, spindles are attached. They will ask you the question. In metaphase 1, the particularly spindle fibers are attached to centromeres, centrosoles. Then they will give you third option, kinetochore or anything else, or all of this. So answer is for you. If you observe this figure, you can see that they are attached to centromere only, but there is one structure which is present just like a block-like structure close to this centromere. We call that structure as a centromere, and outside structure we call it as a kinetochore. So these spindle fibers, these spindle fibers, they are where they are attached? They are attached to our kinetochore, which are present close to the centromere. So अभी क्या हुआ? प्रोफेज में, प्रोफेज 1, डोंट गेट कंफ्यूज. मियोसिस में प्रोफेज 1 में हुआ रिकॉम्बिनेशन, जेनेटिक एक्सचेंज हो गया, गॉट इट. उसके बाद हो गया, वो सेपरेट हो गए. रिकॉम्बिनेशन होने के बाद टर्मिनाइजेशन उसको बोलते हैं. मेटाफेज में आके क्या हुआ कि वो सेपरेट होने के बाद भी वो होमोलॉगस क्रोमोसोम दोनों सेंटर में आके लेट गए. बोथ ऑफ देम came and get arranged at the all the chromosomes. Just I have given you an example one. All the chromosomes will come and get attached to the centromere. What it get attached to the centromere. Now we'll see the next point. This is all about metaphase 1. Now after metaphase, which phase is there? Anybody can tell me which phase is there after metaphase? Which phase will come after metaphase 1? Anaphase 1. What is Anaphase 1? I have told you for that one. That is PMAT. You remember PMAT? P means prophase, M means metaphase, A means anaphase, and T means telophase. You just remember the word PMAT. हमको ये word ध्यान में रखना है. PMAT means you will know that what is the sequence. So this is all about metaphase. Now we'll see the next phase, that is anaphase 1. Now we'll see the next phase, that is anaphase 1.
Madam, it is a centromere splites and three sister chromatide which chromosomes separate. They pulled apart opposite direction with the help of spinal fiber. It's called as a sister chromatide, was called a daughter chromosome.
डॉटर क्रोमोसोम, वो ये हमारा अगली प्रोसेस में होता है. दिस विल ऑकर फर्दर. व्हाट इट? वेट वन मिनट. Now we'll see the next stage, that is anaphase 1. We'll see the next phase, that is anaphase 1. Now what happened, this anaphase 1, you see here, we'll see the figure afterward. You just first see, and afterwards I will show the figure, and again I will explain. So what happened there? So first we'll see the figure. Let it be. So what happened in this anaphase? You can see this is anaphase and this is telophase. Anaphase में क्या होता है? That is all the chromosome fiber contrast. First क्या होता है? Chromosome fiber contrast and pull the homologous and interchromosomal fibers extend so that homologous chromosome start moving towards the opposite pole. यहाँ पे आपने देखा है, ये देखो, ये पूरा रेड वाला क्रोमोसोम, इसके 4 क्रोमेटिड से wait 1 minute. You can see there are 4 chromatids, 1 2 3 4, इसको बोलते हैं हम bivalent. Got it? Now you can see genetic exchange has occurred between that particular seed. Suppose this one, इसका रेड पार्ट यहाँ पे आ गया, उसका ब्लू पार्ट यहाँ पे आ गया. What happened there? Because genetic exchange has occurred between them. उनके chromatids में gene exchange हो गया है. Genes have exchange between that particular chromatids. Now what we'll see, मेटाफेज में क्या हुआ था कि ये सब इधर सेंटर में आके लेट गए थे. अभी एनाफेज में क्या होगा, that is they start moving towards the opposite pole. Now this 4 chromatid will move towards this pole and this 4 chromatid will move towards this pole. That is one chromosome of homologous pair. Homologous pair का एक क्रोमोसोम, ये कब होता है, जेनेटिक एक्सचेंज ऑलरेडी हो गया है, वो सेंटर में आके मेटाफेज में लेट गए. अभी उनको क्या करना है, उनको सेपरेट होना है. दोनों को क्या हुआ है, काम हो गया है हमारा जेनेटिक रिकॉम्बिनेशन वर्क हैज बीन ऑलरेडी कंप्लीटेड. नाउ व्हाट, they have to separate from each other. Now you can see in this figure that both this is moving towards this pole and this one is moving towards this pole. That is only the speciality of this particularly this phase, that is homologous chromosomes start moving towards the opposite pole. One thing you remember here, the centromere do not divide. एक बात आप हमेशा ध्यान में रखना, जैसे कि ये एक क्रोमोसोम है, हमने देखा है, this one is one chromosome, this is one chromosome. Suppose, just 1 minute, suppose this one is one chromosome. Suppose this is one chromosome, this is one chromosome, this is another chromosome. ये दोनों का सेंट्रोमियर, ये जो राउंड होता है वो कभी डिवाइड नहीं होता है. ये इधर जाएगा और ये इधर जाएगा. हमको क्या लगता है ये कट हो गया और ये आधा इधर जा रहा है, आधा. It is not like that. Centromere, always this question was asked in 2018, whether the centromere divide in anaphase 1. It is no, the centromere do not divide, just homologous chromosome. Is homologous means what? This both are homologous. This will move here and this will move here. Centromere दोनों का जैसे कि वैसा ही रहेगा, वो divide नहीं होगा. Don't get confused. During this stage, homologous chromosome which are still attached as chasmata finally get separated. और ये जो separation होता है इनका, this one get here and this one get here. This particular concept, it is called as disjunction. What it is called as? Disjunction. Disjunction बोले तो क्या, वो साथ में थे पहले, जंक्शन में थे. जंक्शन बोले तो वो एक दूसरे के पास थे, they were close to each other. Now disjunction, उनका जो जंक्शन था वो टूट चुका है. That is called as opposite one, disjunction. That particular concept we called it as a disjunction. Now next point is, each chromosome is with 2 chromatids. Now you can see each of the chromosome with 2 chromatids. Suppose this one is one chromatid and this is another chromatid. This is another chromatid having exchange segment. और इनके पास क्या है, exchange segment है. उसको what do you mean by exchange segment? You can see here, this one is our red chromosome. फिर भी उसके पास ब्लू का पार्ट आ गया है और ये हमारा पूरा क्रोमोसोम ब्लू था तो उसके पास रेड वाला पार्ट आ गया. Means what? Although they have separated, although disjunction has occurred between that chromosome, but that disjunction chromatid, they have exchange segment. उनके पास क्या प्रेजेंट है, exchange segment of both of the chromatid. At the end of anaphase 1, what will happen at the end of anaphase 1? At the end of anaphase 1, number of chromosome gather at the respective pole. वो क्या होता है एंड की, ये तो स्टार्ट हुआ कि जैसे स्टार्ट हो रहा है और एंड जब होता है तो क्या होता है, दिस chromosome has reached here and this has reached here at the end of anaphase. So now next phase you can see telophase 1. You can see telophase 1. Now what happened? Very simple concept. Telophase 1. What happened in telophase 1 now? You can see that chromosome uncoils to form chromatid. You always remember telophase is just opposite of prophase. जो prophase में disappear हो गया है वो reappear हो गया. Means what you can see here. हमारा nucellus जो round structure था, this one, ये nucellus it was disappeared during prophase. It was reappeared during the telophase. न्यूक्लियर मेम्ब्रेन जो हमारी डिसअपियर हो गई थी प्रोफेज में वो फिर से रीअपियर हो गई. सेंट्रोमियर्स जो नए सेंट्रोमियर्स फॉर्म हो गए, दो here two cells are formed. As you can see, अभी ये at anaphase what will happen? This chromosome will completely come and settle here. This will come and settle here, and two cells are formed. Two cells are formed. Here you can see that is two nucleus are formed. Two nuclear membrane. This one is one membrane. This one is another membrane. This one is one nucellus. This is another nucellus. Here there are two centrioles. Here there are again two centrioles. That is chromosome uncoils to form chromatid. It will form chromatid. The spindle fibers. ये जो spindle fibers, ये lines जो यहाँ पे आपको दिखाई दे रहे थे, वो क्या हो गए? Disappear. That spindle fibers have disappear. As you can see, the appearance of all the cell organelles. All the cell organelles start appearing. जैसे भी cell organelles appear होना शुरू हो जाते हैं, हमारे spindle fiber disappear होना शुरू हो जाते हैं. पूरा disappear हो जाते हैं telophase में. और next point is what is speciality of telophase? That is chromosome become uncoiled, spindle fibers disappear, cell organelles begin to reappear. The nuclear membrane, particularly which, that is nuclear membrane and nucellus will reappearing forming two daughter nuclei. How many daughter nuclei are formed now? Two. This one is one and this is another. Got it? From one cell you have got two nucleus. This is called as mother cell, suppose, and this one we call it as a daughter cells. From one daughter cell, two daughter nuclei are formed. This is the end of telophase 1. End of telophase 1. Students, are you getting what I am teaching? All of you, are you getting what I am teaching? Students, can you hear me? Hello, students. Ayush, can you hear me? Ayush. Kuntal, can you hear me, Kuntal? Okay, we will start now. Next point. We will start now. Next point. That is, now we have already completed meiosis 1. We have already completed meiosis 1. Now we will start meiosis 2. But there is one more process between meiosis 1 and meiosis. Which is that process? We call it as a cytokinesis. उसको क्या बोलते हैं हम? Cytokinesis. This we have already seen. Telophase में क्या होता है? The chromosome uncoil to form chromatids. The spindle fiber disappears. The spindle fiber disappear. The nuclear membrane and nucellus reappear, forming two daughter nuclei. Now, after telophase 1, now which is the first process? आप ये बात हमेशा ध्यान में रखना, जैसे हमने सेल न्यूक्लियस तो फॉर्म कर दिया हमारा न्यूक्लियस नया फॉर्म हो गया, वी हैव फॉर्म न्यू न्यूक्लियस, इजंट इट, फॉर्मिंग टू डॉटर न्यूक्लियर. अभी जो न्यूक्लियस, दैट इज एज यू नो दैट देयर आर टू प्रोसेस, दैट इज कैरियोकाइनेसिस एंड साइटोकाइनेसिस. कैरियोकाइनेसिस में क्या होता है, फॉर्मेशन ऑफ न्यूक्लियस, वी कॉल इट एज अ कैरियोकाइनेसिस. अभी न्यूक्लियस अगर फॉर्म हो गया है तो उसके लिए साइटोप्लाज्म भी चाहिए हमको. जैसे न्यूक्लियस है तो उसके लिए साइटोप्लाज्म भी चाहिए, साइटोप्लाज्म भी डिवाइड होना चाहिए, तो ही दो सेल फॉर्म हो गए. सो टेलोफेज 1 इज ऑफन फॉलोड बाय डिवीजन ऑफ साइटोप्लाज्म. अभी हमारा न्यूक्लियस है वो, उसको भी लिक्विड चाहिए और वो भी सेपरेट चाहिए. जैसे कि हमारा सेपरेट न्यूक्लियर मेम्ब्रेन आ गया है, न्यूसेलस आ गया है, तो हमको अभी सेपरेट साइटोप्लाज्म भी चाहिए. दैट इज टेलोफेज 1 is often followed by division of cytoplasm. You can see the division of cytoplasm. The plasma membrane constrict in the middle in the animal cell, while cell plate is formed in plant cell. Always remember that. ये मैंने आपको बताया, जैसे कि साइटोप्लाज्म जो डिवाइड होता है, whenever the cytoplasm divides, what happens? Whenever the cytoplasm divides, 1 minute. Look here, all of you. Suppose what happened in, what is different between plant cell and animal cell? Animal cell में क्या होता है, ये जो साइटोप्लाज्म डिवाइड होता है, whenever the cytoplasm divides, whenever the cytoplasm divides, you can see, suppose this is our one cell, this is another cell. What happened in animal cell? Suppose this is animal cell. Animal cell में क्या होता है, यहाँ पे ऐसे एक फरो फॉर्म होता है, दोनों अंदर की तरफ खींचते हैं, that furrow is formed, and when this furrow go in, ये both furrows will touch here, and you will see the new cell, it is formed in this way. This is one new nuclei and this is another nuclei. That is, you can see here, furrow is formed, FURROW, furrow. Furrow is formed in case of animal cell. The cell plasma membrane, ये जो plasma membrane है वो अंदर की तरफ जाती है और furrow form करती है, and that furrow when both will reach together, they will, you will see the appearance of new cell here. Suppose this is one cell and this is another cell. इसके opposite में animal cell, plant cell में क्या होता है? What happens in plant cell? What happens in plant cell? Plant cell, that is, suppose this is one cell. Suppose this is one cell. This is our one cell. Plant cell में क्या होता है? At the middle, cell plate is formed. Cell plate is formed. What that cell plate? How is that cell plate? Cell plate is like that. That it is just like this. It is just like this. This one. Suppose this is the cell plate. Suppose this is the cell plate. और यहाँ पे one cell होता है और यहाँ पे दूसरा cell होता है. तो ये cell plate क्या होती है? बाहर की तरफ जाती है. It will move towards outside. It will move towards outside, and finally it will give rise to the two new cells. This is nucellus. This is nuclei of one cell. This is another cell. Means in case of plant cell, cell plate is formed, and in case of animal cell, cell furrow is formed. For what purpose it is formed, or in which process it is formed? It is formed in cytokinesis. What do you mean by cytokinesis? Cytokinesis is nothing but it is the division of cytoplasm. It is the division of cytoplasm. जब cytoplasm divide होता है उसको हम बोलते हैं cytokinesis. जब न्यूक्लियस डिवाइड होता है उस प्रोसेस को हम बोलते हैं कैरियोकाइनेसिस. गॉट इट? नाउ उसमें भी अलग अलग है. जैसे कि प्लांट सेल में सेल प्लेट फॉर्म होती है, वो सेल प्लेट after some time it is converted into cell membrane. It is converted into not cell membrane, middle lamina. It is converted into middle lamina. Or in plants, in animal cell, you can see the furrow-like appearing structure which is moving inside, and in plant cell it is moving outside. Ultimately what happens, that both cells get separated, this cell and this cell, this both cell you will see that they have separated. So this is all about, now we have completed metaphase 1, anaphase 1, telophase 1 and cytokinesis. Now remaining meiosis 2 is very easy. If you properly understood meiosis 1, meiosis 2 is very easy. Although students, if you are not understanding this, we are all going to repeat these sessions when you are coming offline here. So don't worry about that. I will again repeat these sessions. Now we'll start the new point, that is meiosis 2. Meiosis 2. You just see this figure. You will get light idea what happened. What happened in meiosis. This figure, if you understood this figure, you will come to know what happened actually in my meiosis. Now, suppose we were having diploid cell. ये कब होता है diploid? ये जो particular stage है, suppose this one. This is the particular stage. This stage, when you get this in prophase. Prophase में क्या मिलता है आपको? Sorry, you can see this phase in S phase. You can see this in S phase, that is interphase. अभी ये दो homologous chromosome है, that is one from paternal chromosome and one is maternal. How this diploid 2n, how it is formed? This is N, maternal side. This one is N. This one is N. This is N. This paternal side, it is also N. And this both, it will form 2. That is 2n. It will form 2n. Got it? After what happened? Duplication of chromosome. Yes, phase के बाद क्या होता है? ये single chromatid होता है इसको. You can see here, it is having single chromatid. You can see here, it is having single chromatid here, like this. Suppose this is one chromatid, this is another chromatid. Duplication of chromosome after S phase. S phase के बाद वो क्या होता है? S phase, it is present in interphase. Interphase, you all remember, in interphase there are 3 phases: G1 phase, S phase and G2 phase. S phase में क्या होता है? Duplication of chromosome. After duplication, what will happen? Suppose this is our original. After duplication, what will happen? You will see the appearance of new chromatid here. This one will be new. So you can see here also. This one is blue वाला है, उसके साथ में blue वाला नया तैयार हो जाता है red वाले के साथ. Means in this way. That is, this one is already present from maternal side and paternal side. And duplication के बाद वो क्या होता है? इसका ही this one, this blue is already present and this yellow is duplicate of this blue. Means what? इन दोनों के यहाँ पे जो जीन्स होते हैं वही जीन्स यहाँ पे होते हैं. Got it? Each chromosome had now 2 chromatids. यहाँ पे कितने थे? They were having only 1 chromatid. Here you can see each chromosome is having 2 chromatids. Got it? Now meiosis में क्या होता है? What happened in meiosis? That is during prophase. Prophase में क्या होता है? Genetic material का exchange होता है. You can see the change in color. That is, some part of this chromosome is present here and some is present here. That after the exchange of genetic material, after the recombination, you will see that in meiosis 1, these 2 chromatids or these 2 chromosomes get separated. These 2 chromosomes get separated. We have just seen that. We have just seen that process. That is called as meiosis 1. That is, यहाँ पे जो 2 थे homologous, इनके बीच में exchange of genetic material हुआ. प्रोफेज में पैचिटिन स्टेज में एक्सचेंज ऑफ जेनेटिक मटेरियल और दोनों सेपरेट हो गए, गॉट इट, उनका साइटोप्लाज्म भी सेपरेट हो गया। अभी मियोसिस 2 में क्या होगा, ये जो दो क्रोमैटिड से ये जो स्ट्रक्चर है, यू कैन सी दिस स्ट्रक्चर, you can see this structure. Now suppose this is like this, you can see this structure, suppose this structure is like this. Now what it, now what will happen in meiosis 2, what will happen in meiosis 2, ये बीच में से ब्रेक हो जाएगा, दो क्रोमैटिड्स इधर जाएंगे और एक क्रोमैटिड इधर जाएगा, एक क्रोमैटिड इधर जाएगा, ये दूसरे इसका एक इधर जाएगा, एक इधर जाएगा। In this way 4 new haploid cells are formed, 1 2 3 and 4, just they will centrally get divided, ये सेंटर से क्या हो जाता है, ये डिवाइड हो जाते हैं, एक इधर आ जाता है, एक इधर आ जाता है, ये one will come here and one will come here. In this way from these 2 chromosome you can get a 4 haploid cell. We have already completed till meiosis 1. Now we'll see meiosis 2. This is all about meiosis 2. Now meiosis 2 में भी अलग अलग stages होते हैं. That is meiosis 2. Actually, it is very short resting period between meiosis. One point is very important. There is a very short resting period between meiosis 1 and meiosis 2. ये जो meiosis 1 है यहाँ पे और ये meiosis 2 है, इसके बीच में in the middle of this 2 stages there is a resting period. We call it as a interkinesis. उसको क्या बोलते हैं हम, interkinesis. That particular period this question is asked, interkinesis, that is called as interkinesis. That is short resting period between meiosis 1 and meiosis 2. We call that period as a interkinesis. It may or may not be present. It is not like that it is always present. In some cells it is present and in some cells it is absent. Now this meiosis 2, it consists of you just take screenshot of this. That is diagrammatic representation of meiosis. It is very important. You just read one more time in your textbooks. You will come to know this point very clearly. So what happened in meiosis 2? Now tomorrow we will take that point, that is meiosis 2, but we'll just what happens actually in that. This meiosis 2 consists of 2 substages. It consists of 2 substages, that is karyokinesis and cytokinesis. I have already told that it consists of 2 substages. Karyokinesis it consists of 2 substages, karyokinesis and cytokinesis. Karyokinesis and cytokinesis. Karyokinesis means what? Division of nucleus. Now that karyokinesis consists of different stages, that is prophase 2, metaphase 2, anaphase 2 and telophase 2. Students, don't get confused. We have started meiosis process. In meiosis there are 2 processes, meiosis 1 and meiosis 2. We have completed meiosis 1. What happens in meiosis 1? There are different stages, that is prophase 1, metaphase 1, anaphase 1, telophase 1. After telophase there is cytokinesis 1. In metaphase 1, prophase is converted into liptotin, zygotin, pachytin, diplotin and dikinesis. Got it? Now in meiosis 2, meiosis 2 consists of same stages, but just as it is second time dividing, so it is called as 2. That is prophase 2, metaphase 2, anaphase 2, telophase 2. Got it? Now both the haploid daughter cells are formed at the end of meiosis 1. Meiosis end में क्या देखेंगे आप? कि हैप्लॉइड डॉटर सेल्स फॉर्म हो गए, हैप्लॉइड डॉटर सेल्स फॉर्म हो गए, एंड एट द एंड ऑफ and it undergoes meiosis II simultaneously. Why it is told simultaneously? At one time टू हैप्लॉइड सेल्स आर फॉर्म. ऐसे नहीं होता है कि एक हैप्लॉइड सेल डिवाइड होता है फिर दूसरा रुक जाता है, ऐसे कभी नहीं होता है. दोनों एक साथ डिवाइड करते हैं. यहाँ पे one side one haploid cell will divide and another side another haploid cell will get divided. In this way you can see that meiosis 2, it consists of prophase 2, metaphase 2, anaphase 2, telophase 2 and last one. अभी 4 फॉर्म हो गए तो उनको साइटोप्लाज्म भी अलग चाहिए, तो फिर से उनमें क्या होता है, साइटोकाइनेसिस. दैट इज साइटोकाइनेसिस 2. that cytokinesis 2, what will happen in that cytokinesis 2? That is cytoplasm will get divided. Cytoplasm will get divided. So remaining part we will see in the next lecture. Okay students, any questions? Any questions? Any questions students?
Anh nói có thể sẽ tôi gọi là